Alright, now let's move on to our next topic, which is how pH affects enzyme activity. Enzymes work best at a certain pH, and we call this their optimum pH. This is the pH level where the enzyme is most active and works the fastest. Every enzyme has its own optimum pH, some prefer acidic conditions, while others work best in neutral or alkaline environments. At this optimum pH, the enzyme's structure is stable, and the active site, which is the part that binds to the substrate, has the perfect shape. This is because the R groups on the amino acids in the enzyme have the right charges, which helps maintain the enzyme's proper 3D shape. But when the pH moves away from that optimum, either becoming too acidic or too alkaline, the enzyme starts to lose its shape. This is because the change in pH affects the charges on those R groups, and that can disrupt the enzyme's tertiary structure, which is its 3D shape. Once that happens, the active site doesn't fit the substrate properly anymore, and the enzyme doesn't work as well, or might stop working altogether. You can actually see this on the graph here. The enzyme activity rises as we get closer to the optimum pH, reaches a peak, and then drops off sharply if the pH gets too high or too low. That drop is because the enzyme is becoming denatured, in other words, it's losing its shape and function. So to sum it up, enzymes need the right pH to work properly. Too much change in pH can break down their structure and stop them from doing the job. Let's explore it a bit further. So, what exactly happens to enzymes when the pH changes? Well, altering the pH can sometimes cause denaturation, which is when an enzyme loses its proper shape and, as a result, stops working correctly. Remember, enzymes are very picky, they function best at a specific pH, known as their optimum pH. Different enzymes have different optimum pH values depending on where they work in the body. For example, the enzyme pepsin, which breaks down proteins in the stomach, works best in very acidic conditions, around pH 2. That makes sense because the stomach itself is a very acidic environment. Now compare that to amylase, the enzyme in your saliva that starts breaking down starch. It doesn't like acidic conditions at all, it becomes inactive at pH 2. Instead, it works best around neutral pH, which is about pH 7. Inside most of our cells, enzymes also prefer this neutral pH. But in the duodenum, which is the first part of the small intestine, the environment is slightly alkaline. This suits enzymes like pancreatic lipase, which breaks down fats and works best at around pH 8. It's also worth noting that small changes in pH can affect enzyme activity, but these effects are often reversible. For instance, if an enzyme temporarily stops working in a low pH, it may become active again once it returns to its optimum pH. Let's look at the graph provided here, to visualize this. The x-axis represents pH, from 0 to 12, and the y-axis shows the reaction rate of the enzyme. You'll notice three curves here. The red curve, labeled pepsin, peaks sharply at pH 2, showing its high activity in acidic conditions. On the other hand, the blue curve, labeled pancreatic lipase, reaches its highest point around pH 8, indicating its preference for alkaline conditions, whereas, the third curve represents most of the enzyme's preference, which is working in a neutral condition. You can also see a color-coded pH scale from 0 to 14, red for acidic, green for neutral, and purple for alkaline. This helps us connect where each enzyme works best within the body. So, overall, this graph highlights a key idea: enzyme function is highly dependent on pH, and each enzyme is finely tuned to operate in the environment it was designed for.